Hello everyone, welcome back to the Genomics Bootcamp and the final part of the Introduction to Genomics series. This presentation will be about genomic selection. As always, we start with a little bit of summary from the previous lectures. So we continue to talk about SNPs, so the single nucleotide polymorphisms. We also continue to talk about the so-called SNP chips that cover the whole genome, and we analyze the data based on this. And one of the returning topics, the linkage disequilibrium, or the LD, that characterizes the degree of relationships between nearby loci and SNP markers. Because we have this dense SNP chip data, which covers the entire genome, then we have dense enough markers to have the entire genome covered. This means that we can be sure that one or more SNPs are in high LD with any given part of the genome. And this information will be instrumental and extremely important to understand the genomic selection. Well, the genomic selection is to improve certain traits, certain phenotype. And if you think about it, it might ring a bell to you because it is actually very similar to GWAS. In the genome-wide association studies, we had the genotype and the phenotype. And our goal was to identify the regions on the genome that influence certain trait. This is of course possible, and eventually we could find the region on the genome that influences this trait, and we can learn something about the biology of the organism or how the genomic regulation of the trait is working. One difficulty, however, is to detect genes that have a really small effect. And actually, these are the ones that we are mostly interested in when we want to improve the traits, especially the production traits of the animals. So ultimately, we are after identifying these locations, these quantitative trait loci, or QTL in short. Now, in general, the quantitative traits are very important in animal breeding. Almost all production, reproduction, and functional traits are quantitative traits, which means that they are influenced by a high number of genes, with each of these having a very small effect. So in other words, we cannot pinpoint one or two locations on the genome and select just for those and expect an increase in the productive traits. But instead, we have an unknown number, possibly thousands of places, which each contribute with a small amount. Well, as I mentioned, there are many quantitative traits, but to make it easy and make it very straightforward, I will use the classic example of the milk production. Well, the most important role of animal breeding is, of course, food production. We need to consistently improve the populations so we can sustainably produce food that fulfills the needs of the mankind. Of course, the animal products are not the only source of food, but they greatly contribute to achieve the goals and targets highlighted by SDG 2, Zero Hunger, and this is of course related to the Sustainable Development Goals of the United Nations. So we have a great question. How do we select individuals with thousands of genes, and all of these genes are on unknown locations, and on top of that, with each of these genes contributing only a tiny proportion to the trait? So how can we select the best animals based under these circumstances? And the answer is relatively straightforward, using genetics. The improvement of the livestock populations is not a new thing, and in fact is being done for long decades. Here is how a traditional breeding program looks like based on progeny testing. I will introduce this program first and then contrast the genomic selection to it. These breeding programs are purely based on records, on statistical models, and do not involve any genomics. And as I mentioned, I will use the milk production as an example. So at this first level, there are the parents. There are very good cows and sires, and basically they are mated together to produce, sons. I have to mention here that in animal breeding, the male individuals are much more important from the genetic standpoint, because while the cows could have only relatively few offspring throughout their lifetime, constrained by the biology. The males can have hundreds, thousands, or even tens of thousands of offspring because of the artificial insemination techniques that are used throughout the livestock breeding and especially in cattle breeding. So basically, this whole selection process is focused on bulls. But there is one big problem with bulls, and this problem is that they don't produce milk. So if our goal happens to be the improvement of the milk production in the population, we are in a little bit of a problem. Of course, there is a way out of this, and that is to measure the milk production of the daughters of this particular bull. So this is what happens here. So basically, these young bulls are mated to the so-called test population or a few cows, well, let's say 100 or so. And then these offspring are born and the daughters grow up. They are mated as well. And after their calves are born, they start to produce milk. And after the first lactation, we have the level of the milk production of the daughters of this particular bull. And then we have our phenotype, which we can insert into our models, and also considering the pedigrees and all the relationships within the population, we can construct something which is called a breeding value. And based on this breeding value, we can decide if this bull is good or not. And if it's good, then we keep it and use it to a wide scale mating throughout the population. Eventually, there will be some very good cows as well, and everything goes back to square one here. And we have another generation of bulls that is then better than the one before it. A bit of explanation to these numbers here. So these stand for reliability. So this is the reliability of the breeding values that are computed. This system, as I mentioned, this is a traditional system, and it's proven to be working extremely well. As I mentioned, is being used for decades, and throughout this time, it has proven to increase the production levels of livestock populations by a great amount. A bit of a drawback of this system is in this, well, we could call this a y-axis, the time. It takes actually a lot of time until we can decide whether this bull here is good or not, and if we could use it throughout the population. That is because it itself needs to grow up, then needs to be mated, then these calves need to be born, also grow up, mated themselves, the calves being born, and then after that, there is a 300 days of lactation that is being measured, and then is the time point where we have actually our phenotype. This whole process can take 6 years or more. As I mentioned before, there are sophisticated statistical models running in the background, and this is the so-called Bayes linear unbiased prediction, or BLUP. This is just a short version of it. So basically this Y value, so these are the vector of records. Let's say these are the milk production from daughters. We can have various fixed effects. We have a random effects, that is the animal itself, and then we have an error term. Also, very importantly, there is the relationship matrix, the so-called A matrix, that is based on the pedigree relationships within the population. So all of this is based on written records and does not involve any genomics, but it's proven to be working. It involves a lot of matrix algebra and all that stuff. So this is the short version of it again. But basically, we have estimates of the fixed effects and estimates of the random effects. And basically, these estimates of the random effects are the EBVs, or estimated breeding values. And we use these EBVs to make decisions which sires and ultimately also dams are the best. And these are then mated together and producing an on average better follow-up generation. Of course, we can ask the follow-up question if we can do better than this traditional system. And the answer is yes, we can use genomics.
Now, the use of genomics in animal breeding is, well, not new. And, well, there was this so-called marker-assisted selection. I mentioned this here in this video just to clarify what it is and to underline that the marker-assisted selection, or MAS in short, is not the same as genomic selection. So these two are two different things. It is an older method that, to my knowledge, is not used anymore. Now, in marker-assisted selection, there were two possibilities. One, that markers were used to trace the inheritance of chromosome segments from parent to offspring. And the other was that markers were used as proxy for QTLs. So markers were approximately the QTLs, suggesting that they were in linkage disequilibrium with each other. Well, the idea of the marker-assisted selection was okay-ish, but it had a few major problems. The first one being that there were relatively few effects estimated, and these markers were actually not the QTLs themselves. It was difficult to find markers that were in high LD with any QTLs, and there was also this, well, uneasy feeling that the selection is perhaps for the entirely wrong thing, because these linkage phases could be, well, different in different populations or different families. Also, of course, depending from the distance of the marker from the actual QTL, the LD might change and lead to wrong selection decisions over time in different families, in different populations. And ultimately, we can do the exact opposite thing that we intend to do. So to summarize, the idea was good, but there were a lot of unknowns which we could not control. At some point, still, it was really popular because it was easy to use, easy to market. Everybody was happy that they were using genomic information, and it was marketed quite heavily. But all of this changed with the introduction of the so-called genomic selection, which is the current state of our technique for breeding value estimation in livestock. Here, we estimate breeding values based on SNP effects, and this is possible at any time when DNA is available. So this means that we can estimate breeding values right after the animals are born. We have high-density genome-wide SNP chips in all the species. Well, the 50K SNP chip here is mentioned as an example. It happens to be around 50, 60K in most of the populations, which are heavily used for genomic selection. But of course, there could be also other SNP chips. The costs are reasonable, or I would call them reasonable. It's about 20 euro per chip, well, give or take. So the data is not entirely free, but very much affordable for larger breeding organizations. And right here, I would like to use the opportunity and talk a little bit about my personal stance and personal notes on genomic selection. I feel it is important because I keep getting basically very similar questions from people who basically are exposed to genomics, and at the same time they are managing certain populations, be it cattle or other livestock species. Basically, the scenario is very similar all the time. People learn that, well, genomics is a thing, and then basically one of the first questions is, could we implement genomic selection with our breed or with our species? Here, I would like to highlight that from my personal perspective, well, the genomic selection is certainly very attractive and very useful method, but certainly not the only way how to use genomics in animal breeding or plant breeding for that matter. As I mentioned, genomic selection happens to be often among the first questions from breeding organizations, but I feel rushing to it might be a mistake. My personal opinion is that some kind of prior experience is required with genomics before going into genomic selection. Because, and again, this is my personal view, that I see genomic selection as a follow-up step in a process when many steps were taken before. Also, I want to highlight that the genomic selection is not some kind of a magic bullet or a standalone process that solves everything, and it actually needs a lot of supporting mechanisms. So I had the feeling that sometimes students or in general people have the impression that so basically what we can do is come into a relatively secluded place, genotype hundreds or thousands of individuals, and then based on this we can do the the magic of genomic selection, and then we can do genomic breeding values and suddenly improve that population by a great margin. Well, this is not how it works. Genomic selection itself needs a lot of support, needs well, certainly organized breeding, trait recording, routine trait recording, because it has to start from somewhere, and also, well, needs basically people who are tending to it. But back to the idea of genomic selection itself. So basically what we do is we use, well, these SNP chips, and all these SNP markers from the SNP chip without any significance test. As I mentioned, we have a lot of SNPs that cover the entire genome, which guarantees that any QTLs, be it small or large, are picked up by the SNP markers, and they are in some kind of LD with them. So the genomic selection assumes that all the QTL are explained by combination of nearby markers based on the linkage disequilibrium. Based on this genomic information, we can do a breeding scheme that looks like this, and which is in a way similar and in a way very different to the traditional breeding scheme I showed you before. It actually starts the very same way. So basically we have the very good bulls and dams here, which produce an offspring. But as I mentioned, because we have the genomic selection, we can take, well, let's say a blood sample from this individual, run it through all the procedures, and we can determine a genomic breeding value of this calf. And if it turns out that the genomic breeding value is really good, that means that this calf is a very promising for that population. And because, of course, the DNA doesn't change throughout the lifetime, then what we have to do is wait for this calf to grow up, and then this young bull can be used broadly in the entire population. And the most important aspect of this genomic selection scheme is the time which we gain. You remember in the traditional breeding scheme, this whole process took like 6 years or more. Here we actually just need to wait until this calf grows up. So this is about, let's say, 2 years. And then in 2 years, we can use this bull throughout the population. This factor will be a very important aspect when it comes to the increase of the genetic gain. Of course, when we have these many SNPs throughout the genome, many of these will not be linked to the QTL, but still, effects of all SNPs will be estimated. And actually, we know very well that many of these SNPs are not important at all, but we sort of don't care. Basically, we just estimate everything, so we throw everything into a black box. Well, shake that box and see what comes out. We can confidently do this because we have an assumption that the cumulative effect of these neutral markers will be zero. Of course, everything will be estimated, and some will be slightly positive and some will be slightly negative, but at the end, the unimportant markers cancel each other out and only the important markers remain. This is how it looks like graphically. So we have all these, let's say 50,000 markers here, and effect of each marker is estimated, but these minuscule ones kind of cancel each out to zero. And then only the important markers and marker effects remain. Of course, this could be positive and could be negative, But at the end, we sum up all these marker effects and we have the genomic breeding value. Of course, also the genomic selection is supported by sophisticated models, and we have sort of similar as before. We have, again, the phenotypes, so the phenotype records. We could have other effects, but this part is the most important one, and these are the sum of the SNP effects. And as shown here, we basically sum up these SNP effects that produce the EBV or the genomic EBV or GEBV as a result. And we use this for our decisions. And one of the major indicators for this is the so-called genetic gain or delta G. So this is the so-called breeder's equation. And well, the genetic gain itself could be calculated based on the intensity of selection, the accuracy, the genetic standard deviation. So basically, this is the variability of the population, and divided by the generation interval. The genomic selection itself affects two things. First is the accuracy of the selection. Well, this could be both a positive and negative effect, but this is a relatively minor one because the accuracy is relatively high already. But the major thing is, as I underlined also in one of the previous slides, is the generation interval. Because we can genotype and decide already after birth, we achieve that basically we cut down the generation interval from, let's say, 6 years to 2.5, 3 years. So effectively, what we are doing is we halving the generation interval, which actually doubles the genetic gain, which is huge. These two tables are just to give some numbers from a paper from Sheffer 2006. Well, it's an older paper, but this is just to demonstrate also in numbers what I mean. But also the genomic selection is running for many years now, and very similar results were achieved in major populations. So basically what I meant in the previous slide is the two things, the accuracy and generation interval. So this would be a conventional traditional program. So you see there are 4 categories: sires of bulls, so basically fathers of bulls, and fathers of cows, and dams of bulls and dams of cows. So the accuracy in these 4 levels is, well, a bit different. Well, the sires of bulls are the most accurate, but also they are the most in terms of, or highest in terms of generation interval. If we do all the calculations, we get into a certain 0.2 genetic standard deviation per year. If we go with the genomic selection, so you see here, there is a somewhat lower accuracy for the sires of bulls, while there is an increase in dams of bulls. And most importantly, the generation interval is much, much lower, which leads to a doubling of the genetic gain. So why we expect so much from genomic selection? So in short, it is a total paradigm shift in animal breeding, especially when it comes to thinking about selection. We can produce accurate genomic breeding values of animals that are without records. For example, the young calves that shortly after they birth have breeding values for their milk production. Of course, there are also ways how to do it in a traditional system, but these are usually not very accurate. So we still needed to wait until these animals grow up. In these follow-up slides, I wanted to demonstrate a little bit also from the technical side how the genomic selection is initialized. Also, I hope this will underline my previous notes, why one actually needs some supporting mechanisms, and in particular trait recording when starting genomic selection as such. So the prerequisite of genomic selection is, of course, existence of populations that are both genotyped and phenotyped. So there is a bunch of animals here, and all of these have genotypes, let's say 50K, and also phenotypes for that particular trait that we want to improve or compute genomic breeding values for. Now we divide this population into two parts. The larger part, part A, is the so-called training data set. And part B is a bit special because, well, we delete the phenotype information for this particular set. This is the so-called masking. So basically what we do is we run the models, the statistical models in the part A or the population part A, and we estimate the SNP effects. And based on these SNP effects, we compute the genomic breeding values for population B, or this part of the population B. Now, as I mentioned before, this part of population B is, well, we have genotypes, but also phenotypes that are already measured. So basically what we do is compute the correlation between the genomic breeding values that were computed based on our genomic selection approaches and the phenotypes that were actually measured. And if these correlations are reasonably high and we are happy how it turned out, we can actually use these SNP effects on other parts of the population, including future generations. And this is actually what happens because, of course, after some time, there are new calves born. So this is the part of the population C. And these individuals are only genotyped. So these are the young calves. And we actually have no idea about their breeding value for milk production, for example. But because we have already our genomic selection procedures in place, and we checked that this is working between population, part of the population A and part of population B. So this was okay here. So based on this information, we can be reasonably sure that it will be working similarly well in part of the population C. So basically, we are using the SNP effects that were estimated here and produce the genomic breeding values for these new and young individuals as well. And then we can use these genomic breeding values in our decisions. Well, there is a lot of literature about methods and models that are used in genomic selection, and a lot of them have a lot to do with prior assumptions on marker effects. Well, as I mentioned, there is a lot of literature, discussions, technical details. So if you're interested in this stuff, for sure you find a lot of reading material. For the purposes of this presentation, I make it very simple. So basically there are two large groups. One is based on the BLUP, which is used also in the traditional breeding value estimation, but this is the genomic BLUP or GBLUP in short. And there is then a lot of models and a lot of methods based on the Bayesian approach. So this is the so-called Bayesian alphabet. So this is really a lot of models included in this single bullet point. In papers based on simulated data, some models seem to be working better than others. In reality, however, these differences between the models might not be as huge. So basically, we are speaking about improvement of a few percentages or fractions of percentages in terms of reliabilities and accuracies. Along similar lines, I would like to underline another method that is frequently used, and this is the so-called single-step genomic selection. Previously, when we were speaking about genomic selection, we assumed that all of the individuals are genotyped. Well, on the other hand, especially in large populations, this might not be the case. So there is a part of the population that is genotyped and possibly a larger proportion of the population that is not genotyped. But still, we have all the relationships and we can obtain these relationships from pedigree information as in the traditional system. So this is the so-called A matrix, and we can obtain additional information for the genotyped individuals. So these are from markers, so genomic relationships. So this is the G matrix that is used in the genomic selections. This is used in genomic selection. This is used in the traditional selection. So we have this wealth of information from the pedigree data, but at the same time, we have this supposedly more precise information from the genomic data. Pedigrees in livestock are deep and connect most of the animals, but especially in large populations, only relatively few animals might be genotyped. So the few is, of course, in relative terms, but let's say a lower proportion of the animals are genotyped. And these include mostly the most important bulls and recent animals. This makes us a little bit unhappy because A spans across all animals and G is more precise. So there is a solution that we can actually use both of these sources of information together and join these two relationship matrices, which is actually the single-step genomic selection. So there is a lot of matrix algebra also included here. And so these are quite similar to the ones that I showed you before. So this is exactly the same with the traditional breeding value estimation, and this is the single-step genomic selection. And you see that basically the only difference is the relationship matrix. So this is now the so-called H matrix, which is basically the combination of A matrix and G matrix. And basically, I skipped the details here, but you see that this H matrix, well, this is already the inverse, but still, so this H matrix includes the A matrix and G matrix. So basically, this is the combination. And if you include this H matrix into the overall system, then you can include the animals that are genotyped, but also those animals that are not genotyped, but contributing to the whole system based on their pedigree relationships. And while we have a relatively more complex system in genomic selection, or in this case, just to highlight the difference, so this is the multi-step, but nobody mentions this, but this is just the genomic selection, the classical one. We have multiple steps, so we have basically blood with the records and the pedigree and everything. And then we have the genomic selection part, which could be some Bayes methods or GBLUP or things like that. And then we combine everything into the index, which we then get out an EBV. In the single step genomic selection, we have it much more simple. We have the pedigree records, all the other records, and the SNP effects, and basically the genotypes. We put everything together in a single model, and we get out the genomic, or basically a single step genomic EBVs that we use in our selection decisions. And before we move to the summary of this presentation, I just want to acknowledge the sources that I used for this presentation. So this is a very particular presentation because, I used quite a number of other people's materials to put it together. And actually in the initial form, it was quite a bit longer and also containing more details. But at the end, I decided to cut it down a little bit, so to arrive to this, which I believe a relatively straightforward first information about genomic selection. Still, I would like to acknowledge my sources here, which are basically slides and thoughts and ideas from the following presentations. And that is the marker-assisted and genomic selection from Theo Moivis and Trygve Solberg from NMBU Norway, non-linear model for genomic selection from Theo, application of genomic selection in breeding schemes, also from Theo Moivissen, a relationship matrix combining pedigree and markers from André Legara, INRA, France, also materials from Vincent Ducrot, INRA, France, and forming single-step mixed model equation and quality control from Ignacio Aguilar from INIA, Uruguay. And to summarize this presentation on genomic selection, so basically we are after improvement of production traits that happen to be quantitative. That means that they are influenced by many genes and QTLs of small effect. These QTLs are evaluated based on screening of SNPs throughout the genome, so on the whole genome, and the important SNPs are taken up and the non-important SNPs cancel each other out. So based on this information and based on these SNP effects, we obtain this so-called genomic breeding values. This is a really good thing because the genomic selection and the genomic breeding values could be obtained and the whole system be implemented whenever DNA is available. Of course, considering the supporting factors that also needs to be in place. When implemented, the genomic selection leads to increase of genetic gain because of the decrease of the generation intervals. There are a number of approaches and methods to derive genomic breeding values, most importantly, the so-called GBLUP and the range of Bayesian methods. There is also something called a single-step genomic selection that enables to evaluate both genotyped and non-genotyped animals together. So basically merging the relationship matrices from pedigree and from genotypes, enabling to implement the genomic selection in the entirety of the population. So this was already the last slide, not just for this presentation, but for this whole presentation series. Of course, there is a lot more to talk about, and there are a bunch of other methods, approaches, or ways to use genomic data. I might address those as well in a different series, but for this introductory series, I would like to place the final period here. If you have any questions or comments, of course, put them down in the comment section below. If you like this video or this series, then of course I would be happy if you push that like and or subscribe button, or if you send these videos to whomever you think they might benefit from it. For now, however, I'm happy that it is done, frankly. And I once again thank you very much for your time and wish you an extremely very nice day.